Thank you. Thank you. It's an honor to be here and to present you an update on Asterias Biotherapeutics. Asterias is a publicly traded company on the New York Stock Exchange, and I refer you to our forward-looking statements policies. So Asterias has two transformative technology platforms. The first is a dendritic cell immunotherapy, which actually has two different products. The first is known as ASTY Vac1. It is an autologous vaccine for the treatment of acute myeloid leukemia, and we're in the planning stages for a phase 3 clinical trial to be executed hopefully in the next year. The second program is called ASTY Vacto. It is an allogeneic vaccine derived from human embryonic stem cells that is entering a phase 1/2 clinical trial in partnership with Cancer Research UK. The indication is in non-small cell lung carcinoma. The second platform is our pluripotent stem cell-based platform. And the first example of this is a product called AST-OPC1. These are oligodendrocyte progenitor cells, again, derived from human embryonic stem cells that are being used for the treatment of spinal cord injury. We are now in the process of dose escalating in a second clinical trial in patients with complete cervical spinal cord injury, and we're executing this program in collaboration with the California Institute of Regenerative Medicine. So let's first turn to ASTY Vac1. Again, it's an autologous dendritic cell-based therapy that is pulsed with the tumor antigen known as telomerase. Dendritic cells, again, the potent antigen-presenting cells of the immune system, in this case being used to present the protein telomerase to the immune system to generate immune responses to telomerase. Why telomerase? Telomerase, again, is highly expressed or relatively highly expressed in most cancer cell types. It is one of the hallmarks of transformation into cancer that allows for the continued proliferative lifespan of cells, particularly in cancer. Again, it's expressed in about 90% of cancers. So again, the idea here is to use the antigen presentation immune stimulatory activity of dendritic cells to present telomerase to the immune system to generate immune responses to telomerase and hence tumor cells. We completed a phase 2 clinical trial in patients with AML with ASTY Vac1. In this case, patients with intermediate or high-risk cytogenetics were vaccinated with ASTY Vac1. In this case, the patients were in complete remission, and following a leukapheresis and manufacture of ASTY Vac1, they were vaccinated with 6 primary vaccinations, followed by 6 biweekly boost vaccinations. Patients were followed up for relapse-free survival. So this is the top-line data that we presented at ASCO this year. There were 19 patients who were treated with ASTY Vac1. As of their last follow-up, 11 of 19 patients were still relapse-free. Again, these were patients with both intermediate and high-risk cytogenetics. The median follow-up time was 52 months, so encouraging relapse-free survival. Importantly also, if you looked at patients over 60 years old, of which there were 7 patients enrolled in this particular trial, 4 at a median follow-up of 54 months were still relapse-free. So these data compare very favorably to historical relapse-free survival in these patient populations. And if you look at patients over 60, in general, only 10 to 20% remain relapse-free as of about 4 years of follow-up. So again, this data was very encouraging, and particularly in this indication where there is a high unmet medical need. There are no good therapies for patients over 60 years old due to the comorbidities of current therapies and transplant. So in this particular case, we saw a favorable profile not only for administration to patients, but also in relapse-free survival. So where we are right now is developing our plans for a phase 3 registration trial. We're working with our clinicians, we're working with FDA, we're working with manufacturing specialists to put together the whole package that is going to be required to engineer and to execute this phase 3 clinical trial, which we anticipate will start in the first half of 2017. So let's move to AST Vacto. Again, AST Vacto is a second generation product. In this case, it's the same as AST Vac1, but the dendritic cells now are derived from human embryonic stem cells. So they are an allogeneic dendritic cell platform pulsed with telomerase. Again, the objective is to use this platform to drive immune responses to telomerase and hence to tumor. We're particularly excited about this platform because it uses human embryonic stem cells. Because of the properties of human embryonic stem cells and their scalability, what you can see is that we can use manufacturing schemes that are very similar to that of traditional biologics in the pharmaceutical world today. So you can make large master and working cell banks of embryonic stem cells that can be differentiated in batch to form these dendritic cells, cryopreserved and then distributed at will to patient caregivers. So again, this type of system using the embryonic stem cells allows scalable manufacturing in the allogeneic setting that can be used to supply material to for patients in very large indications. We're working together very closely with Cancer Research UK to take this platform now to the clinic. In this case, in patients with non-small cell lung carcinoma. We're looking at a clinical trial that it will have 3 different cohorts. We'll be using Vacto to immunize and vaccinate patients who at 2 different stages of non-small cell lung carcinoma. Patients who have been resected and have no evidence of metastatic disease, and also patients with advanced disease. And the objectives really are to look to see, is AST Vacto safe? Is it safe in both the settings of resected and advanced cancers? We're also looking at, can we generate immune responses to telomerase? And are those telomerase immune responses active? And to get some initial insights into the activity of these particular cells, and particularly in patients who have advanced cancer. So the results of these studies will really serve as a launch pin to look at further, deeper studies in non-small cell lung carcinoma, but also other indication, and potentially with other immunomodulators. The last product I'm going to be talking about is AST-OPC1. Again, an example of our pluripotent stem cell technology. AST-OPC1, they're oligodendrocyte progenitor cells derived from embryonic stem cells, again, allogeneic, and they have a characterized composition of cells. These cells have many properties, such as that they produce neurotrophic factors, they can induce remyelination of axons, they can induce vascularization. All features that you heard from the last speaker can be useful for repair of spinal cord injury. Again, the intention is really to use this as an off-the-shelf product. Again, the first indication in spinal cord injury, where there are over 12,000 new patients every year just in the United States, and where there's a lifetime costs of patients with high cervical injuries of about $8.5 million a year. So we initiated a phase 1 clinical trial in patients with complete thoracic spinal cord injury. That trial is now complete. We found that AST-OPC1 was well tolerated. There were no SAEs associated with the immunosuppression we use along with the cells. The cells themselves are the surgery. We found that there were no immune responses generated against AST-OPC1. We saw some hints of activity, even at this low dose of cells that we gave in this clinical trial, suggested we had an impact on preventing cavity formation in these patients. and that we saw no adverse effects in terms of neurological function as a result of implanting these cells. We have now worked with the FDA and with our clinical investigators to begin a dose escalation trial, in patients with cervical spinal cord injury, complete cervical spinal cord injury, where we feel that there's better chances of seeing activity and efficacy, and where the outcome measures for improvement in neurological functions are tighter and more quantitative. So we have initiated our clinical trial in patients with cervical spinal cord injury. We have, it's a 3-dose cohort trial, patients in cohort 1 receiving 2 million cells, cohort 2 10 million cells, and cohort 3 20 million cells. We've completed enrollment in cohort 1 right now and are in the process of reviewing all of that data is a prelude to dose escalating to cohort 2. We believe that cohort 2s and 3 are at the dose cohort where we believe, based on our rat studies, where we should start seeing efficacy. So again, we've completed enrollment in cohort 1 and looking at dose escalation to cohort 2 later this year. I should mention that that work was done in collaboration with the California Institute of Regeneration, who is partially funding this particular clinical trial. So I think I will end there, but acknowledge the wonderful team that I work with at Asterias and our senior executive team, Pedro Lichtinger, Katie Spink, and Ed Worth, all who have led to numerous contributions to the company. Thank you.